Hello everyone, welcome back. I hope you had a good spring break. So I made an announcement maybe 2 weeks ago, and I said that your midterm was going to be due on the 10th, yesterday, but that's actually during spring break, and I felt kind of bad, so technically, I changed it to 4/17, so if you haven't done it already, make sure to do it. You're going to have 30 questions in 20 minutes, so it's going to be pretty tough, but I think you have plenty of time to get it done. Just make sure you're prepared. You've studied labs 1 through 7, you know everything from the PowerPoints and the midterm reviews, and I'm sure you'll do fine, okay? So make sure you get that done by next Friday. There's also going to be a few other things due on Friday. So there's going to be your midterm, but then there's also going to be your food safety and news discussion board post. So here I have a document which has all the instructions for that assignment. So just read up on that. And then you're also going to take a look at the discussion board. And I have an example post there for you to take a look at in case you're still not sure what to do after seeing the assignment. So there's another thing I need you to do. There's the food safety project instruction, which is going to be your new major assignment for the entire semester since we're not doing our usual food report for the actual lab. This is going to be more of a lecture, a digital assignment since we can't do the lab portion, unfortunately. So here's a template for that assignment, and then this is an example. This is one that I created a few years ago. So if you have any questions about that, feel free to email me. What I want you to do this week, what is due, is your discussion board post and then also the midterm. But in your discussion board post, you need to make sure that you answer all the questions in the food safety and news document, and then you also are going to include your food product that you're going to be writing about for this food safety project right here, okay? So read those instructions, figure out your food product that you want to use for your food safety project, and then write about all that in the discussion board. So if you go to the discussion board, you'll see that there's one post made. So what I want you to do for your title is the organism involved in your outbreak, when it took place, and then I want you to put the name of the food product that you're going to use, but not the one that's involved in your outbreak, but the one that's involved, the one that you're going to be using for your food safety project, okay? So the one you're going to be using for that assignment. And then you're just going to follow this format that I've laid out right here, okay? So, if you have any questions about that, feel free to ask me. But this is just a little example that I made. Feel free to read it. So, with that, that's going to be everything that is due. And then I also want to go over this lecture so that next week I'm going to be posting the Lab 9 lecture, and then I'm also going to have a quiz on Lab 8 and 9 that you'll have to do. So I'm going to go over this right now. If you have any questions, email me, and I'm happy to answer them as soon as I can, okay? So, last lab, in quotation marks. I'm doing it with my fingers, but you can't see, obviously. So, what we should have done is we would have been looking at our Baird-Parker auger and our Biochrome SRES auger. So what we should have seen is if it's going to be Staph aureus, it's going to have these kind of these halos around the colonies. So it's going to be these black colonies with these wider margins surrounding them, like opaque zones, clear white zones around the colonies. And then for epidermitis, you're not going to have the clear zones around them, and sometimes they won't even really grow at all. They'll just have a few colonies at all, maybe no colonies at all. So with the Biochrome Staph aureus auger, then we're going to see the Biochrome plates are going to change color depending on the organism, so they're going to be differential. So the Staph aureus are going to have mauve colonies, kind of the purpley, pink-ish color, and then the epidermitis are going to have no color, or weaker growth, okay? So again, we're always going to be more concerned with the Staph aureus because this is your pathogen. The epidermitis is not going to be dangerous, so it doesn't really matter if it doesn't grow. Or we want to be able to tell these apart because we want to know if we have Staph aureus And we don't really care too much about staph epidermidis, okay? So, for Lab 8, we're talking about bacilli and Clostridia. So bacilli, you should know, is the shape, kind of the rod shape for organisms. So, as the name implies, they are rod-shaped. These are also going to be gram-positive bacteria, so this is the genera Bacillus. They're going to be spore-forming, aerobic or facultative anaerobic, and then they're going to be living primarily in the soil, so they're going to be soil organisms, but then they can also be found in feed, manure. So this is going to be causing a lot of contamination for food and plant products, since they are outside and a lot of our food does come from the soil. And it's going to be causing a lot of spoilage and foodborne diseases once we eat those organisms if it's contaminated a food product. So the first one is Bacillus subtilis, which is going to be aerobic, found in soil. I mean, most of these are, but this is going to be a really good organism for studying sporulation. So when we do spore stains, this is a very common organism for that. And it's going to be causing food contamination, okay? Major food contamination organism. Then we have B. cereus, our facultative anaerobe. This is another great one for spore stains, as you can see here. So spore stain right here, we have the green, malachite green for the spores, then the pink out here, dyed with the safranin. So these are going to be commonly found in soil, and then they're going to be producing toxins. These toxins are going to be causing symptoms when ingested, which is going to cause you to have diarrhea, nausea, and vomiting. Attempt to expel these toxins from your body. So it's a really serious one with mishandling your food, kind of heat abusing it. So you cook it and then you leave it out for a really long time, and then that gives a chance for the for the spores to turn into vegetative cells and then reproduce, and then you can inform more spores and more toxins, and then when you eat the food you'll get sick. Even though you may boil it, it's not going to kill the spores, unfortunately. So then we have Clostridium, which is our next genus. So these are going to be gram-positive bacteria, rod-shaped, spore-forming, anaerobic only. And then they're going to be found in soil and also intestinal tracts, okay? So ingested. So these can cause some gaseous spoilage of foods, as in C. perfringens, and then also they can cause some pretty severe foodborne illnesses. As you've probably heard of C. botulinum, right? It causes botulism. So Clostridium perfringens, this is anaerobic, and it's going to be found in soil and play a very prominent role in decaying vegetation. And then also it's going to have an involvement in the human intestinal tract, be a part of the human microbiome, and also some insects. But it's going to be producing enterotoxins that cause intense abdominal cramps, gas, and diarrhea. Responsible for bacteremia, emphysematous cholecystis. Oh boy, that's a big word. I don't like these words. And gas gangrene. Oh, glad I'm not a doctor. So, this is not an organism that you want to have as a part of your normal microbiome. This is a pathogen, so it's going to be causing some serious problems, okay? So, Clostridium botulinum, which is first isolated in 1896 by van Emmingem. So it's going to be associated with food poisoning, particularly for canned foods, honey and sausages. So this is a really big problem for infants, especially in honey. So that's the main reason why infants can't have honey. So it produces botulinum neurotoxins that cause botulism, which is a serious paralytic illness. So 10 grams of this toxin is enough for a lethal dose for everyone living in Los Angeles. In order to work with it, you actually need to have a special permit and be clear to work with it. So Clostridium sporogenes is a closely related non-pathogenic strain, okay? Sporogenes is going to be our non-pathogen for this. So, quite a bit more serious than Clostridium perfringens, but RC Clostridium sporogenes is going to be not a threat to us at all, basically. Non-pathogenic. So, for today's procedure, we are going to be doing some real fun stuff, except we're not doing anything, so basically just testing how these different organisms look on these different plates. So, observing the morphology of these. Oh boy, fun. Okay, so yeah, so then we'd be using boiling water to test the heat resistance different times, but we're not going to be doing that anymore. As you'd expect, though, spores can survive exposure to boiling water more than vegetative cells, just in case you didn't know. Okay, so with that, that's just about everything I wanted to cover. Just make sure to complete your midterm. Okay, it's online. Everything's online, just based off of Labs 1 through 7, that midterm review. And then we also have this PowerPoint right here, which is going to be a part of that quiz next week, based off of this lab 8 and 9, just the lecture part. We're not the lab portion. Like, I'm not going to test you on this stuff, okay? And then make sure to do your discussion board forum post for the news project, that assignment, or the food safety news. And then make sure to put your food product in that discussion board post, okay? Make sure we know. And with that, that's everything.